before we hear a bit more about fire, I'm going to talk a bit about understanding animal adaptations in changing environment. And here's a list of some of the people that have worked with me on these kind of problems. Environmental changes come in different forms. There are natural changes like seasons and so forth, and anthropogenic changes like climate change. I spent my career studying things like seasonal changes, and I contend that you really need to understand those sorts of adaptations, the animal's ability to change, the flexibility to change with the changing environment naturally, before you can even start addressing questions about anthropogenic changes. The changes, or the kinds of adaptations that animals have include long-term evolutionary changes that involve genetic adaptations, but within the lifetime of an individual animal, there's also the plasticity, the possibility of plasticity, both behavioral changes, behavioral shifts, and physiological shifts. And a lot of the work we've been doing is looking at the limits of this flexibility, in particular with respect to the thermal environment, but also the hydric, or the moisture in the environment. And sorry, this was a picture of a local frog in a cocoon, and they can survive in this kind of a state for several years. So this is a very, very unique adaptation that an animal has to survive a changing environment. First project I want to talk about is geckos in refugia, and where we look, and this is in collaboration with, it's an ARC discovery project with Dr. Craig Moritz, Professor Craig Moritz at ANU. We're looking at geckos across northern Australia that have a couple of different kinds of distributions. Some of the species live in very small ranges inside of refugia, those refugia being areas that have had, typically around permanent water, but they've had long periods of climatic stability. And then the more widespread species, and there are two sorts of widespread species, some of which live only outside the refugia, so in the savanna sea that surrounds these refugia islands. And then there are some species that live both inside and outside. And so that natural experiment, those different combinations, are very good, very powerful tool for answering some of the questions that we want to ask. And the kinds of things we're asking are about the population genetics, that's Craig's work, and then my work is on the physiological responses of these different groups of animals, and looking at the plasticity as well as species differences. In particular, we're looking at the thermal environment, the thermal adaptivity, and their hydric. So the question, the title of the project is, are refugia evolutionary traps for specialized species? In other words, have these species that are living in these kinds of specialized environments evolved more narrow tolerances? And does that mean that they are more sensitive to climate change? These are very fundamental, basic questions of evolution and ecology, but they provide the baseline information for management or conservation decisions, things about how important are refugia and how do we conserve both specialist species and generalist species. The second project I want to talk about is looking at the skin microbes on animals. The microbes on the skin of animals have been shown, this is a very sort of emerging field of research, they're shown to be important for a variety of reasons. Probably the most important is that they're in disease resistance. We're looking at some of the ecological relationships in this area, because the microbes on the skin of an animal, some are from the environment and some are actually promoted by the skin. And this is important in the context of climate change, because the distribution of diseases will change with climate change, as well as the fact that hosts can become stressed in changing environments. And a lot of basic information is missing about this ecological context of the skin microbes. Some of the results, or recent results, is, first of all, there are species-specific microbial communities. So we have, here on campus, we have 3 very closely related species living exactly in the same environment. They have completely different microbial communities on their skin. We've also found very recently that what was thought to be very much of an amphibian characteristic, is actually also very important on the skins of reptiles. A couple of data slides are showing relevant to diseases. The slide on the left shows that the two sites, Tully and Innisfail in Queensland, where chytrid fungus is very important amphibian skin disease, is prevalent. And the toads, cane toads in those areas have a different set of microbes that specifically inhibit the growth of fungi, and chytrid is a fungus, as compared to toads in the Northern Territory, that's Middle Point, and also further to the Western Australia. So there's very different microbial communities there related to that disease. Even more recent information shown as this graph on the right, where we look at cane toads, the susceptibility to a lungworm. The lungworm enters through the skin of the cane toad, so the skin microbes are important. The orange dot or yellow dot on the right shows the natural state of a toad's skin with its intact microbial community. And on the left, the blue dot shows what happens if you disrupt that microbial community by swabbing the animal and cleaning it a bit. It's 3 times more susceptible to the lungworm invasion. These lungworms have come with cane toads to Australia first from Hawaii, and then to Puerto Rico, from Puerto Rico to Barbados, and then French Guiana. So the lungworms have traveled with the toads, and so it's not unreasonable that it's possible that some of the microbes have also come with the toads, and that's what one of our future projects is looking at, the resilience of this skin microbiome with respect to these toad translocations, these intercontinental movements of toads. Have they brought their external immune system with them? And also the other, related to that, is that this lungworm system is very manipulable with respect to experiments, and so we're going to attack the problems in that way. Finally, very, very quickly, one last project that we're looking at, the orange-footed scrubfowl mounds. Those mounds are, the microbes in the mounds provide the heat for the incubation of the eggs of these birds. And so the question is, well, how does that change in a changing environment? And this goes back again to my first point, that looking at how important, or what is the ability of animals to mediate the environmental changes through their behavior. And that's one of the things we'll be looking at. Thank you.